Hello AP Bio students, so today we're going to talk about biostatistics, and I know this all seems a bit overwhelming and like a lot of math, but it's actually not too bad because I'll break it down. And compared to AP Chemistry or AP Physics, I think the math in AP Bio is a lot easier. So we can start with the basics with some descriptive biostats. We can start with the 3 M's, mean, median, and mode. So you've probably heard of these before, maybe in elementary school you were taught them. And I don't know about you, but whenever I'd learn these, I'd always confuse them because they all start with M and they all have pretty similar definitions. So I came up with mnemonic devices for you to remember them. So the first one is the average person is mean. So when you're finding the mean, you're finding the average. So you're basically adding up all the data and then dividing it by the number of data points or the number of observations. So basically it's a type of average. And mean is also represented by this symbol right here. It's called X bar. I'm going to be using that symbol a lot throughout the video, and just know that it means mean, which is a type of average. So the other M is mode. So another mnemonic device would be the current mode or fashion is what we see most. So in a data set, the mode is the most repeating number. So if I have 114, that's my data set, then the mode will be 1 because that's the most repeating number. And then the last one is median. Well, I couldn't really think of a mnemonic device for this, but the median is the middle number. I guess they both have 6 letters, so I guess that's a way to remember that. So those are the basics, but you really need to know mean the most. It's the most important one out of the 3 for AP Bio. And so now we're going to talk about standard deviations. So I'm going to represent standard deviation as S, sometimes SD. So standard deviation basically tells us how spread out our numbers are from the mean. Basically tells us the range of our data. So if we were to graph our data and if it were to look something like this, and let's say we have another data set and if we were to graph it, it would look something like this. Sorry, that's kind of bad. This would have this data set right here, since the graph is kind of more spread out. This would have a higher standard deviation because it means that our data is more spread out. The first one, the data is not as spread out. It's kind of clumped together closer to the mean. So it's pretty like stuck together in like a herd. But in the data point where the standard deviation, in the graph where the standard deviation is the greatest, the data is all spread out. There's probably like a lot of outliers and it's a huge range. So standard deviation basically tells us how spread out our numbers are from the mean. So I'm going to talk about how to calculate standard deviation. So here's our formula. I know it looks kind of intimidating, but it's really not too bad. Also, for the AP Bio test, this might vary from teacher to teacher for your unit tests in your AP Bio classes, but I know on the AP Bio test, this is given in the formula sheet. And if you haven't looked at the formula sheet for AP Bio, and if you're taking the AP Bio test, you should definitely look at it, because there's a lot of important formulas like standard deviation, this one. There's also the water potential formula and some other ones that you should know. So I definitely would recommend checking that out. So in the standard deviation, we take the square root of each data point and you're subtracting the mean from it. So this x means a random data point, and then you're subtracting the x bar or the mean from it, and then you're squaring it. And then once you do that for one data point, you have to do that for all the data points and then add them together. And if you're confused by this, I'm going to show you examples so it's going to make more sense. And this symbol means summation, so you're adding everything together. So this means that you're adding everything together. And then you're dividing it by the number of data points. So let's say we have 113. Then our n would be 3 because there are 3 data points, and we're subtracting 1 by it, and then we're square rooting it. So the reason we square this is because we want it to be a positive number when we add them together, and then we kind of square root this to kind of unsquare the square, if that makes sense. So here is a graph of a normally distributed data set. So it kind of has this like really nice bell curve. So when a graph has normal distribution, not all graphs have this. This is just certain like ideal standard perfect graphs. 68% of the data will fall within 1 standard deviation. 95 will fall within 2 standard deviations, and then 97.7% of the data will fall within 3 standard deviations. So you kind of see that here. So here's 68% of our data. So 68% of the data. And then it all falls within 1 standard deviation, which we can calculate from this formula. So plus 1 standard deviation or minus 1 standard deviation. And then right here, all over here, we have plus or minus 2 standard deviation, and that encompasses 95% of our data. And then 99.7% will fall within 3 standard deviations. So probably like this, the rest of it. So why do I need to know this? Well, many statistical tests, like the t-test, you don't need to know that. That's for higher level college courses. Rely on the fact that the data follows a normal distribution. So the data is relatively like this perfect bell curve that we see here. The data is not too like spread out or sporadic, or else that will kind of like mess up our analysis. So the reason we do biostatistics is to help us derive the meaning from our data. So if you're like pre-med or pre-health or just like gonna go into biology in college or just majoring in life sciences, you're probably gonna have to do some sort of research or labs in the future. And when you do research, you probably have procedures that you follow, and you have a question and then you have procedures that you follow to try and answer that question, doing your experiment, collecting the data. So once you have all your data from your experiment, you just have all these numbers and you don't really know what it means. So that's why you run all these statistical tests on it to kind of analyze your data to tell you what your data means. Does my data support my hypothesis? Does it reject my hypothesis? What do I need to know for my conclusion? So we run statistical tests. We use biostatistics to basically give us meaning from our data, tell us what all these random numbers mean. So in college, you'll probably use coding to do like a lot of statistical tests. You probably won't have to do a lot of it by hand, but it depends on your professor. So many statistical tests, like the t-test, which you don't need to know, but that's just an example of a statistical test that you can do, rely on the fact that data follows a normal distribution. So our data can't be too spread out or crazy, or else that'll kind of mess up our analysis. And if your data is too wild, then there's a good chance you probably messed up when doing the procedures, like human error. I'm not sure. So the normal distribution shows how data is evenly spread out around the mean. So we saw this kind of like perfect symmetrical bell curve. I know my drawing's not perfect, but it's supposed to be like pretty nice and like a perfect standard ideal kind of data set graph. So, I forgot to show you this, but here is an example of calculating standard deviation. So right here. So here's my tiny little data set, and it's 5 points. So our n is going to be 5, so we have that right here because it's n minus 1. And then here's each data point, so that's our x, and then we're subtracting the mean from it. That's our 5, that's our x bar. So you can see that here for each point we're doing that, and then we're squaring it. And then since this means to add it all together, we are going to add them together, and then we square root it. And then if you do everything correctly, hopefully you should, because on the AP Bio test you should have a calculator, so you just kind of punch it in. You should get this number. So why do we need to know standard deviation? What is the point of all these random numbers? Well, you're going to use this formula during your conclusion. So in your conclusion, after you do a lab, you're going to talk about how your data confirmed your hypothesis. You're going to talk about your analysis of your data, and then whether it confirmed your hypothesis or whether it taught you to refute your hypothesis. And it's also going to show you how strong your data is. And you do this by calculating the standard deviation. Obviously, if your data is all like super spread out and all over the place and it kind of looks like this curve, yeah, your data might not be very strong. There's a good chance there's a lot of human error when you're doing the lab. But if your data is like pretty like together, kind of like this, then your data is probably really strong and we're doing the procedures correctly, possibly. So basically, you need to calculate this in your conclusions for biology.